In this video, we shall be talking about the primary structure of proteins. We shall discuss how these primary structure of proteins are formed, what is an amide linkage, how the polypeptide chains are formed, and how these polypeptide chains combine together to form more complex structures that give rise to secondary, tertiary, and quaternary structures. So now let us discuss the primary structure of proteins. A primary structure of protein is the particular sequence of amino acids. It is a linear chain of alpha amino acids. They form the backbone of the peptide chain or protein. The free amino group of the terminal amino acid is called as N-terminal, and whereas the free carboxylic end is known as a C-terminal end. You can see here is an example of a polypeptide chain in which cysteine, tyrosine, phenylalanine, glycine, asparagine are linked together to form a single peptide chain. So now let us study how these peptide chains are formed, how a peptide bond formation is there. We shall understand this by taking an example of a peptide bond formation between valine and alanine. Now let us draw the structure of valine first. Now this is the structure for valine. As we all know that it is an hydrophobic or a non-polar amino acid. Now let us draw the structure of valine. So this is the structure of valine. It will be forming a peptide linkage with that of alanine. Now let us draw the structure of alanine. We will be using a different structure, a different color to represent the alanine molecule. So this is the structure of alanine here. A peptide linkage will be formed between the N-terminal of alanine and C-terminal of valine. This results in the formation of a dipeptide by removal of a water molecule. Thus it gives rise to a new dipeptide known as valyl alanine. Now we shall be drawing the structure of valyl alanine here. Now we shall be using a different color to represent the amide linkage. So this is the amide linkage here, and thus rest is the structure of alanine. Now this compound is known as valyl alanine. Now this is how a peptide bond is formed. This pink colored bond is known as a peptide linkage or a peptide bond. Now we shall be discussing an example of insulin here. Insulin was the first protein to have its own primary structure determined. It has a primary structure of two polypeptide chains linked by disulfide bonds and has a chain A with 21 amino acids and chain B with 30 amino acids. So this was just an example of a polypeptide chain in insulin. I hope the structure of valylalanine is clear to you all, and you all can now easily draw a peptide bond formation or a peptide linkage. This is the most easiest structure in the proteins. In our next class, we shall be discussing about the secondary, tertiary and quaternary structures. So till then, revise the peptide bond formation, and you can take examples of more polypeptide chains. You can also draw the structures for a tripeptide by combining 3 amino acids together. So students keep practicing and learning these.